Can we hear me this time? Start this again after the 10-minute rant I made with no sound. Okay, so, right. This is stem cells part 2. The first stem cell talk I did was to understand what stem cells were. This one is where we get our stem cells from. I'm going to go straight into it because I feel like I've been doing this twice. I talked about the difference in potency. Now, potency is a cell's state. It's how differentiated it is, how much of its cell purpose it is. And I talked about the fact that when a cell's fertilized, right up to when it's replicated up to being 16 of them, right, so very few, talking tiny, 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 they're too fragile to take from. And that's a reminder because we never take from that state ever, and that's what I'm saying to the person that I talked to in the comments. It's after that we take cells. Now, they rightly pointed out, when do they ever leave enough cells behind for the cell to be able to start again? And of course I said that that was possible, but yes, you're right, they never do that. In any establishment of a new culture of embryonic stem cells, they always end up destroying the embryo itself. They end up destroying the fetus as it is, of all 16 cells. They can never just take one or two, and it's because of the successfulness of the experiments. They need to be able to repeat the experiments, and they don't want the experiment to fail. Of course, with things like that, you only get, if you're taking a human sample, you only get a certain number of opportunities to take the embryos. And that was in the beginning, though. And so this video is going to explain what we now do, and the fact that the blending babies discussion has to end because it's no longer relevant. I'm sure that those worried about the implications of killing a possible human life won't take anything I say about a baby only being a baby when it's established in the womb, a baby only being a baby when it can sense what's around it. I know people won't take those arguments, so I'm not even going to go into that. We're going to go into why it's no longer valid to talk about the immorality of stem cell research. It's just to put minds at ease, I guess. We have new techniques now. So basically the next thing they tried was that some adult cells have plasticity. They can be changed by chemicals to exhibit different potencies, but they may not actually be those potencies. Like, as soon as you take away the chemical, they react as they normally did. But with the chemical's presence, basically certain reactions are inhibited and things like that. And it's very specific, and it was very difficult to work with. And then the only cases where it actually was successful was where the cells actually came from the person who the treatment was intended for. Of course, at first they did it with mice. In all the examples, what they did was take it with mice. But of course, when they started to apply it to humans, it wasn't the same. The advantage is actually with using that method is that you don't have to use immune repressants. Like when you give a person an organ, they have to suppress that person's immune system so they don't reject those cells. And it was the same with this. Like, you wouldn't have to do that because the cells were the same. So it's kind of like a slight advantage. But there was a lot of disadvantage, it being that it couldn't be applied to all the things that stem cell research could possibly be applied to, because they weren't really that potent. They had to be kept in the chemical containment when they were being made, basically, to make this new tissue set. What they did with one of the cases, a woman with tuberculosis, they gave her a new trachea, or at least the lining of the trachea. They kept the cells in this chemical compound, basically, and they grew, multiplied, and became basically this part of the throat, and they managed to replace it in this lady. But it's still, they couldn't give you a completely new organ and things like that, and they couldn't like replace nerve cells at this point. But now they can do that. The next thing, of course, was iPS cells, which is of course the future, and the new capabilities since 2009 with them are the way of stem cell future. iPS, induced pluripotency, anyone who watched the last video knows what I mean by pluripotency. They're typically made from an adult cell that has already differentiated, and basically the potency, de-differentiating it, making it go backwards, is induced by genetic modification. So you're taking some of the genes that are present and swapping them for different ones. Not the greatest. I mean, people who protest against GM crops, I don't understand their arguments always, but there are hazards, and it's these hazards that these people are going to get worried about. Messing around with genes in that way can make them resistant to certain things, and when you make one thing resistant, its opposition, or its enemies, if you like, the bacteria and stuff, will also become resistant in new ways, and they'll find new ways to integrate, and sometimes those can become so difficult to deal with that you can't. So genetically modifying things can get a bit hazardous, a little bit. Now they have, since 2009, completely eradicated that as well, because now we don't even have to genetically modify them. What we can now do is another kind of setting it in chemicals kind of basis, but this time, once they're put into the protein and they're set backwards, you have to keep them in a specific kind of stem cell culture that was established back in the beginning when you were using stem cells, but they don't have to be kept in the chemical once they've been induced, basically. And it's a type of protein, and it triggers genes that are already there. What partly helped this development was mapping the genome and just understanding the genome and triggering certain things and working it out, basically. So that's why it was only as recently as 3 years ago that we managed to do this. But it's brilliant work, and basically that's the way of the future. What they now do is they trigger it by its own genes, the genes that are already present, to go backwards, and it's from adult differentiated cells. So it's the way of the future. It's going to cure so many different things, or it's hoped to cure many sort of different things. We can give people new organs that belong to them, basically. And so many different diseases in the brain are going to be able to hopefully be reduced in a way. Their symptoms will be reduced in a way, but that's for the theorizers to give you, and I'm supposed to be giving you what's real. So I hope I've done that. So yeah, we've never been blending babies. Before, what we were actually doing was stealing mice fetuses. And there was a few human examples where they tested it out, but there weren't many cases. It was mostly mice that we did this on. So the moral ramifications are over. I always say, if you allow the science to continue as it would naturally, it'll come to good things. And there will always be legislation that stops us from doing certain things. There's issues with cloning and things like that, but that's fine. We understand. If you think we're a different species, no, we're not a different species. We do get it. We'd never clone a human, things like that. So, hope this helps anyone with their stem cell arguments, as always. Happy blending babies.